Aloha, my name is Sam Champine, and for my study, I chose transfer of arsenic and phosphorus from soils to the fronds and spores of arsenic hyperaccumulator Pteris Vittata and 3 non-hyperaccumulators. The study was conducted by Jason T. Lessel et al. The study mostly sought to track the accumulation of arsenic in spores as opposed to tissue and roots, which has been done widely in other studies. Pteris vittata is known as a hyperaccumulator of arsenic, so it's often chosen for these type of studies. However, the study did measure leaf tissue arsenic levels as well as root tissue, and they measured the arsenic levels in the soil. To cross-compare, 3 other ferns that were thought to be non-accumulators were also measured for their arsenic levels. These ferns were Thailipteris kunthii, Nephrolepis falcata, and Nephrolepis brownii. Basically, they divided the study into 3 sections. So they had one section which were natural ferns grown in clean soil. So these were ferns that were not cultivated. They were found in the wild. They chose these groups because they had a good portion of the population that was mature, and the ferns were harvested at peak spore maturation so that they could get the arsenic levels tested for all the spores. The second section was natural ferns in arsenic contaminated soil, and the last section was a group of cultivated Pteris vittata ferns they also did in contaminated soil. One thing that I feel they left out of the study was doing a cultivated test on the other 3 ferns. However, I do believe the way that they went about their methods in this study were very well done. It was very straightforward, but a lot of information, obviously a long read. They basically took the leaf tissue samples from all of these groups. They took the root samples and they took the spore samples, and they ran the leaves and roots through sieves, and then they mixed it with a nitrate, a nitric acid, and then they digested it using a hydrogen peroxide solution, and this was able to separate out the arsenic as well as phosphorus. So the study not only measured the arsenic but the phosphorus as well, and as the authors explain, the reason behind this is because arsenic is often uptaken with phosphorus. Arsenic is not known to have any nutritive benefit to plants, and so one of the main theories is that it is just being taken up with phosphorus. However, ferns that do not seem to accumulate this are thought to have maybe some sort of blocker that will stop it from uptaking phosphorus and arsenic so that it can avoid the toxic effects. However, the toxicity is not noted in something like Pteris vittata, which hyperaccumulates this from the soil. So I thought it was a good paper. They took all of these results and they compiled them, and they found some pretty interesting results. Here's a chart showing all the different rates of arsenic which were found in the sites. This is the arsenic concentration with standard air. I did find the standard air to be rather large, and I'm not exactly sure why this is. The authors don't touch on this very much, and so perhaps this could be an issue in the study. However, they do put together this matrix which shows significant values, and so I thought the statistics on this were pretty good, and it was a very well conducted study. Moving on to the results, which were very fascinating, they found, of course, the expected correlation between a positive correlation between the roots, root growth, or sorry, root arsenic level, leaf arsenic level, and spore arsenic level with the soil arsenic levels in the contaminated soils for Pteris vittata, as well as the non-hyperaccumulators. And the non-hyperaccumulators accumulated more than they were thought to have. For the clean areas, it was a bit of a surprising result in that they found lots of accumulation. Actually, the highest rates were from the clean soil for both Pteris vittata and the non-accumulators. This was a bit of a surprise, and the authors suggest a few different reasons for this. One may be that the ferns are just cutting themselves off after a certain level of arsenic uptake, and so when it's in very high amounts in the soil, it cuts off from hyperaccumulation. And one of the other very interesting results was that the spores were found to have extremely high rates of arsenic, and one of the theories as to why this might be something that is beneficial to the ferns and has lasted as an evolutionary trait is the authors suggested that perhaps the spores are not benefiting themselves from the arsenic, but that they are releasing arsenic during germination, and that is perhaps toxic to competition species. The way that they had tested this too was to mix some deionized water in with the spores, and I believe they calculated the arsenic levels that came off in the deionized water. It's kind of a simple experiment, but it gave some stability to that claim that potentially this was causing the competition to have toxicity. However, much more research needs to be done before we can really draw conclusions as to why these ferns are accumulating arsenic under these conditions. So that's pretty much it. The purpose for these studies is mostly to recognize that arsenic could be carried in spores, and that could be a potential hazard to human health. And I believe they don't talk about this very much in the study, but it could have a lot of bioremediation purposes too, to better understand how these ferns accumulate toxic metals such as arsenic. So that concludes my study. This was the first study to investigate the transfer of toxic arsenic and nutrient P levels from soils to roots. There's a lot more small details and information, so I will attach the article to the video. Thank you.